Welcome, everyone, and thank you for joining us today. I'm Candace with the Digital Medicine Society, and I'll be moderating today's panel discussion co-hosted by the Digital Medicine Society and Metadata. We have a great conversation in store for you. Over the next hour, we're going to dig into a pressing question in cardiopulmonary drug development, which is how do we measure whether patients are truly getting better? And are the tools that we've been relying on for years, are they still the right ones? Today, we'll be exploring cardiac effort as a real world example of how wearable sensors are pushing that conversation forward. We'll talk about the data science behind it, what it takes to validate a new endpoint, to what clinical researchers and trial teams can actually do with it today. We are excited to be co-hosting this session alongside Metadata. This is a partnership that brings together cross-industry experts to bridge emerging innovation with real-world evidence. Bringing people together for these types of conversations is central to what we do here at the Digital Medicine Society, and there are plenty other ways to get involved, from convenings to thought leadership to case studies to advisory work. And we hope this session gives you a good sense of some of the ways in which you can engage with our community and help move the field forward. Okay, now to the fun bit. Today's webinar is titled Beyond the 6-Minute Walk, Unlocking Cardiac Effort as a Novel Endpoint. And thankfully, we have the right people in this virtual room to have this conversation honestly. But before we get to that, just a couple of housekeeping. Today's session is being recorded, and as always with Dime webinars, the slides and the recording will be available on our webinar site. Second, participants are not permitted to transcribe this webinar. And finally, there are a couple of ways to engage during this session. During our live Q&A at the end of the last 10 to 15 minutes of this webinar, kindly either raise your hand and a moderator will unmute you so you can ask your question live, or secondly, feel free to type your question into the chat box and we'll try to surface that question to the panel. Okay. Without further ado, let's introduce our panelists. I'll start with you, Melissa, from the top.
Thank you, Candace, and thank you, Dime, for hosting and preparing this webinar for us today. My name is Melissa Cerullo, and my background is in medical devices. I have over 20 years experience in development and commercialization of medical grade devices. The last decade focus on wearable sensors and data that comes off those sensors. And then for the past few years, been focused on developing new endpoints for clinical research and specifically for cardiac effort. Thank you.
I'm Dan LaChant. I'm a pulmonologist at the University of Rochester. My research focuses on wearable devices, coming up with new metrics to detect kind of how patients are doing in clinic and at home. I've been doing this for about 10 years now, and the biggest portion of my research is focused on this cardiac effort of evaluating heart rate changes during prescribed activities as a better metric for how patients are doing clinically.
I'm Gabriel Nalathambi. My background is in wearable medical devices and validated algorithms and how they can support endpoints that clinicians can trust. So that wearable sensor and algorithm perspective is what I hope to add to today's conversation.
Hi, my name is Dmitry Kalikovich. I have a background in digital health. I basically have worked in this field for 8 years, supporting activities related to digital health technologies across the board, including cardiovascular diseases. And these activities include study ideation, event evaluation, selection of different endpoints, analysis of data, as well as interactions with the regulators.
Thank you. Thank you, Dimitri. I'm Very excited to get into the topic at hand today. But before we do, let's open it up for a audience poll just to get some of our juices flowing early this morning. So the question I want to pose to our audience today is, in cardiopulmonary trials, where do you see the greatest unmet need for better functional measurement? And so we have a couple options here. One, reducing variability and noise in existing endpoints. Two, detecting treatment effects earlier or in smaller cohorts. Three, enabling remote or decentralized assessment. Four, capturing the patient's experience of functional capacity. And if there is another unmet need that you can think of, feel free to type it in the chat. All right, we're getting a nice distribution of answers here. Um, it seems like all of these responses seem to be, an unmet need in cardiopulmonary measurement, and some of these topics we'll discuss further during today's webinar. Interesting. Okay. Very interesting. All right, so let's get right into the discussion. We'll start by setting the stage a little bit. The 6-minute walk test is a test that we most of us are familiar with, and it has been an anchor for measuring function in cardiopulmonary trials for many, many years. And so the first question I have for this group is, from your vantage points, where does this test fall short in telling us whether a patient is truly getting better? And as a result of that, what does that cost clinical trials and what does that cost patients? Melissa, why don't I start with you?
Sure. So you're right, Candice, that this test has been a standard for decades. In fact, I think 50 years since its inception. And it really hasn't changed much over the past few decades. We're still doing the test in the clinic in a supervised setting with an unobstructed walkway, and we're still measuring distance walked. With all the advances of technology, we really haven't adopted any, new types of measurements during the test. And so I think one of the first issues is just that supervised setting. Uh, I've actually recently spoke to a patient that did a walk in the Boston hospital area, and she said that the it's very difficult to find a hallway where there's no traffic, right? She's walking around people that are moving along the hallway. And so that's a very simple challenge where the hallways are not always unobstructed and there's sometimes a lot of people walking through. And you need a very long course in order to comply with the ATS standard. Um, so that's kind of the basics. Number two is that there's a lot of variability in the distance measurement. And that can be due to a lot of different factors. That can be due to how the test is administered. That can be due to how the patient's feeling on that particular day. The fact that we're measuring one parameter distance, and that's the endpoint of the study, really leaves a lot of opportunity on the table to miss some other important information about how that patient did the walk. One thing that we've seen in our research is that there could be two different patients that have completely different clinical status. They could walk the same distance, but how they perform and how they function during that walk is very different. And Lastly, I'll mention that it's very difficult to distinguish between physiological changes and fluctuations in effort when you're just capturing the distance parameter.
Interesting. So we've heard about the limitations in supervised setting, variability in administration, some of the limitations with just distance captured. Um, Dan, I want to turn to you since you've spearheaded a lot of the work with cardiac effort. How does this measure um, sort of solve some of that limitation or at least address some of it?
So I've been working on cardiac effort with my mentor, Dr. Jim White, for about 10 years now, and we've been discussing this idea about effort and how much someone puts into a walk and kind of building on what Melissa was talking about. Anyone who was at the American Thoracic Society conference this past year during some of the pulmonary hypertension clinical trials, it was really interesting to see when you looked at 6-minute walk cap clinical trials versus clinical trials where patients weren't capped, the placebo group in advanced outcomes decreased, which is what we would expect in a PH clinical trial at 6 month follow-up. Whereas when you looked at the seralutinib as well as a Merck pooled analyses, the placebo group actually increased. And so that just goes into, well, how much effort went into that baseline walk? Did the participant do it? They knew they had to be below 450 meters. Therefore, they didn't tax their system as much. So they had more reserve. 6 months later, they do the walk again. No one's worried about it. And therefore, their walk increases 15, 20 meters. And that throws off a lot of the statistics that goes into building these expensive clinical trials. Well, cardiac effort solves that because at baseline, you know what somebody's effort is. If they walk 470 meters and their heart rate's 150, well, that tells you that person gave every effort they had in that walk. They're not going to pull a fast one on you if they're in the placebo group and increase by 20, 30 meters 6 months later. And this also allows for further walking patients to get into clinical trials. I think everyone has to recognize, especially in PH and other disease states as well, that we've got highly effective therapies for many different patients. So therefore, the 350 functional class 3 person is more a connective tissue disease, especially in North America. So if we really want to get real world patients into clinical trials, we need to have a way to assess that, yes, this person is functionally, physiologically limited. They would benefit a drug. And we're going to push the bounds in terms of who we allow in clinical trials. And by interrogating the heart rate changes during the 6-minute walk test, it really says, how is that cardiopulmonary efficiency? How is the system working at baseline? And if it makes you feel better in terms of a signal to noise measurement, that who's going into this trial is who you want, and that if they're going to benefit from the drug, it's going to be apparent.
And then just for the audience, Dan, can you describe what the patient actually experiences from receiving the wearable to completing the test for cardiac effort?
Yeah, we've had high patient uptake in our own clinic, and we've deployed it in three, well, one of our own multi-center trials, but 2 pharmaceutical clinical trials have also deployed it. And study coordinators like the technology. They think it's very interesting, and it just puts a different lens, especially younger study coordinators who are very tech savvy. They like it. Patients like it because, one, if we gain more information and we can understand more about what's going on, they're all for it without having to undergo further invasive testing. It takes a minute to put on and set up and get going. And some patients, they feel like they're validated in terms of what they tell us. When they say, I have chest palpitations or I have chest pressure, and we can see their heart rate's 150, 60, well, that says, you know what, this is what the provider team can actually see what I'm experiencing. Other patients, we've been able to pick up atrial arrhythmias during the 6-minute walk test that otherwise would have been missed without heart rate monitoring. And then even in a subgroup of patients, not in clinical trial research, but just doing this measurement, patients learn that they can actually do more safely by seeing what their heart rate does. A lot of what they do is driven by what their care team tells them. And if all they're looking at is a walk distance in isolation without any other information to go along with it, they're going to kind of handicap, tie themselves down, and not move as much as they could because they don't have the reassurance that, yeah, your own heart rate only goes to 115, 120. It's safe to do more. And therefore, they feel better, do more, and everything can then continue to improve.
Great, thanks, Dan. And maybe Gabe, I'll turn this question to you. So we have 2 inputs, heart rate, distance walked. can you describe how we get from that raw sample level signaling to a single value, including, the practical piece of counting beats during a structured walk test and even, motion artifacts during a walk in a home setting?
Yeah, definitely. I think the quality of the output is as important as the value of the endpoint. So here the equation is very simple, as you mentioned, but that doesn't hide the difficulty of measurement. So in this case, we need to define exactly the 6 minute window. And during the 6 minute window, we'll have to identify beats during motion. So that may be false positives, false negatives that we'll have to deal with. We'll have to determine the exact distance for that specific time window, and we may need pre-specified quality rules to ensure that we get good estimates. So any timestamp mismatch, even a few seconds of artifact that could lead to false positives or false negatives, and any bias in distance estimates can completely change the ratio. So it's extremely important that the quality of output is prioritized to get accurate endpoint.
Great, thanks, Gabe.
And I just
Uh,
wanted to
yeah.
add to Gabe's response is that there's not a lot of devices that we can get a good raw accelerometer signal with good ECG signal. And in order to do the calculation as Gabe described with precision, you really need to put the sensor on the chest location. And so we can do a proxy type of measurement on the wrist with PPG, but the kind of the purest method that we've defined is really with a chest patch or a chest monitor. There are a few devices that we've been using, and we are trying to be device agnostic in the algorithm, but the input data is very critical and important, and we've validated and tested across different device platforms to ensure that we're getting that reliability.
Great. So cardiac effort is captured during a structured walk test at home, when done at home. And so I have a pair of questions for this group. Um, that do you see novel measures like this one replacing the 6 minute walk test, 6 minute walk distance test, or complementing traditional in-clinic assessments often used in cardiopulmonary trials? And then the second question, which is related, is what do you think of the role that of measures that capture function in a structured test like this, like cardiac effort? What role do they play alongside measures that capture behavior continuously in daily life? Dimitri, I see you smiling, so I'll let you kick it off for us.
Um, Thanks. I don't know, probably I could have a very long discussion, but unfortunately we are limited in time. So first of all, I like the distribution of the survey you basically presented at the very beginning, but I would rank the answer slightly differently. For me, the top one is to know the variability in the target measure of interest, right? Because the lower the variability, the smaller the sample size. The lower the variability, the greater sensitivity to change of that measure. The greater sensitivity to change, the shorter trials we can run. So, are you aware of a very detailed and comprehensive report of the 6 minute walking test? I'm not. This is a terrible test to begin with. Even though there is a standard MCID, there is a very standard approach to administering this test, but how accurate it is and how well it reflects patients' functional capacity is still highly questionable. Also, if you think of clinical trials, how often or how many times do you typically administer a 6 minute walk-in test throughout a clinical trials? 3, 4, 5 times? But now think of the cardiac effort or any other novel actigraphy endpoint that can be administered in home instead of in clinic. So what stops you to ask a patient to walk for 6 minutes, for example, at home once a day or twice a day. If you consider a typical clinical trial that lasts 6 months to 1 year, this is an arbitrary number, of course. Instead of collecting 3, 5, 7 data points representing the 6-minute walking test, you could collect probably, I don't know, tens, if not hundreds, of 6-minute walking test cardiac efforts measures. And even if the variability is on par with that of the 6-minute walking test, but given a larger volume of data collected, you could better account for the inter-within subject variability, which means that you could better detect your treatment effect if it exists there. Then I'm pretty sure that Dan collected enormous amount of data to properly characterize or evaluate psychometric properties of the cardiac effort. And I'm pretty sure that the variability in that measure should be better than that of the 6-minute walking test, simply due to a much more richer data set or a greater number of confounding factors that Dan can collect compared to the times when the 6-minute walking test was probably collected, characterized, and approved as a signal as a endpoint. So in short, greater as a lower variability, much more frequent administrations, and as a result, greater sensitivity to change.
Dimitri, I couldn't have said it better myself.
Yes.
And the other thing, so we've done a few longitudinal assessments of home cardiac effort, 6-minute walk, and compared it to in-clinic assessment. And some interesting findings that we found is that the 6-minute walk in a modified walking space at home is generally 40 meters less. So that's outside the MCID. So if you have someone do a 90-foot or 100-foot 6-minute walk, and then you have them do it at home on a 40-foot walking space, in general, they're going to walk 40 meters less. When you incorporate heart rate monitoring and you get that effort piece into it, and you got two variables now, you actually get an in clinic and home assessment on a modified walking space that are very similar. They're statistically unchanged with repeated measures. And so it gets to everything that Dimitri was saying is you can get more of these measures over time, which then gives you a signal that says somebody's getting better sooner. You have fewer patients. We've done this on over 140 patients. There's been no safety signal with doing a home unsupervised 6-minute walk test on a modified walking space. We've had 16 to 20 patients on oxygen. We've had high-risk patients do it without falls, syncope, anything like that. So what we really uncovered is you can do a structured assessment that takes the behavioral aspect that actigraphy gives, and it really shows you how somebody's doing.
I would like to add a very simple point. What people tend to forget that most clinical trial data are analyzed using linear mixed effect models. Why is that? Because typically you don't have that many data points per patient. But very important is that the measure or the model assumes that all measures across patients have the same distributions of the errors, which is clearly not the case. Which means that you even shoot in your feet by applying the linear mixed effect models by analyzing 6-minute walking test outcomes. On the other hand, when using a much larger number of data points collected per patient, you can use other methods than linear mixed effect models. So it's not only that you can account better for within subject variability, you could account for potential fluctuations over the course of the clinical trials in functional capacity in that patient. So which again boosts sensitivity to change in that measure.
I'll add here that we've done tests both in both environments, in the clinic as an anchor and in the home environment. And I think that there's definitely value to, both use cases. And one of the studies that we did with the University of Rochester, we added a survey to ask the patients about how their experience was doing the tests at home. And I think the results were really interesting. That first they ease of use of the technology and the instructions that were provided to set up their course at home and to do the test with the wearable sensor. The other thing that was interesting was that they also liked the prescribed push for them to, so at many of these patients, this was their only time that they were doing some exertion or some exercise. And so some of the commentary was like, we liked having a prescribed exercise and we liked having it done in the convenience of our own home. And I think that in between clinic visits, we're seeing that there's a whole new data set, and we had them wear the sensor for a few days and do 2 prescribed walks in the home. And that just opened up a lot more information to understand what's happening in their day to day.
And Melissa, I think that's like consistent with what we're hearing across the field is patients prefer to have these measurements at home versus continually go to the clinic or the lab to complete these tests. But while we're on this topic, how do you handle non-compliance or let's say incomplete home tests in the analysis? How should missing data should be in how should it be interpreted here? Is there anything, maybe Melissa or Dan, that you have to voice?
Yeah, I'll make a quick comment and then interested in the others' responses. So we know, during the protocol design and the planning phase that we need to account for data loss. I think that's just, something that we account for. So um, whether it's because of compliance or non-compliance or just technology issues, in the study design, I think a minimum of 10% data loss should be considered as we're doing this, statistical analysis planning. Um, and then we also need to determine what are the criteria of data quality and how much data are we willing to lose in order to kind of qualify for a good walk test. I think we've had lots of discussions around if we're missing beats because of the noise, then how do we account for that? And can we do some algorithmic um, kind of can we determine what those missing beats are algorithmically, or are we going to just call it a loss for the test in its own? And so I think having these guidelines and criteria up front in terms of what the data quality requirements are is important for us to be able to do a high quality analysis.
I think it's also really important to think about how you're going to deploy this in a clinical trial. What training is going to go into it? What supervision do you need? And to Dimitri's point, if you're going to have multiple assessments and you have people reviewing it real time, and I've worked on some algorithm detections just to say good, bad data quality. You have the ability, because it's in the home setting, for patients to just repeat it the next day. And therefore, you have an opportunity to rescue the data as opposed to an in-clinic assessment. You get your one shot if somebody sprained their ankle and they're down, well, that's it. So when you're developing these protocols, you think about, well, to what Melissa was saying, if maybe there is some data loss from heart rate, is it a patient-specific issue? Is it a device issue? Is it because of a drug? Is it because of X, Y, or Z? And if it's a technical issue, then you have the ability to just repeat it in the home setting and then have another chance at it. And so therefore you really decrease data loss with that ability. Where in a clinic setting, it's harder. You can't necessarily have patients come back, but study coordinator education is key. And working with Liquidia on their Ascent study, they did a phenomenal job deploying this to all the sites. The study coordinators were trained. I reviewed data remotely with them, and they became very adept at good signal quality, and they had very minimal data loss. So between training and just thinking about how you're going to handle certain things is the best way to approach data loss.
Gabe, Dimitri, anything to add here?
Yes. When people say data loss, I'm typically struggling to understand what exactly is referred to. Let me explain what I mean. Does it mean that patient just didn't wear a device? Or does it mean that device stopped malfunctioning while the test was executed? Because these are two very different scenarios. If you think of the cardiac effort or any other digital measures, and those can be collected much more frequently. So let's say every couple of weeks. So in half a year, you will have two measures per month, 2 by 6, 12, versus let's say three measures if you use 6-minute walking test. So if you lose 3 or 4 data points out of this 12, okay, is it a big deal? Probably not. But if you lose 1 or 2 data points out of 3 or 4, that's a big deal. So by definition, more frequent collection of digital measures is much more resistant to patients' not adherence. Then another point, if the device stopped functioning while the test was performed, well, I understand that the target is 6-minute walk, right? But if you could collect 3-minute walk, can't you estimate cardiac effort? Yes, you can. That's not a big deal. Will it have worse variability compared to the measure estimated with 6-minute walks? Yes, it will, but still, you will know the cardiac effort and you will know an associated level of variability in that measure, which means that it can be still used in data analysis. So what I'm trying to say is that much more frequent collection of digital measures in real-world settings is considerably more tolerant to missing data either due to patient not adherence or device malfunction compared to the conventional physical activity measures.
Yeah, I'd also add that, when we're talking about missing data, it may be good to know what we are missing and why we are missing it. For instance, let's say the patch is not in good contact with the skin. That may lead to artifact and maybe false positive or false negative detections. On the other hand, if the patch is not, let's say, within the range of a relay device, then transmission may be interrupted. So knowing the source of the missingness will help us rectify the issue the next time. So that could also help us address, we won't be able to completely make missing data disappear, but I think knowing the source and consequence of the missingness explicit will help us rectify it the next time.
Perfect. Melissa?
Yeah, so you mentioned the 3 minutes, and in fact, we ran a study with 3 minutes to see what the difference is between the duration of the walk, and we're going to continue to investigate if a shorter walk is going to get us the information that we need. And I think so far the results look very promising. This particular study we ran remote. And in the clinic, but we did a full study remote only, and these patients had never done this test before, and so we really wanted to pressure test if we could ship kits out to patients, do a video visit to have them set up their course and training, and then actually perform the tests at home. And we were successful. There was a question in the chat around how to collect 6-minute walk tests at home. And the way that we did it is we put all of the instructions in a patient-facing app. And we also had a clock with a buzzer. So the patients would go through step-by-step instruction on how to put on the wearable device. And then when they were ready to do the walk after they set up their course, they essentially press start, put their phone on in a pocket, and then did the walk in a shortened lap course. They counted their laps and documented the length of their course. So we had that context and metadata in the data analysis. And then they uploaded the data. We looked at the data and aligned that with the timestamps of the start and stop of the walk so that we could easily identify when the walk period occurred. And then we were able to compute the beat by beat measurements, as well as using accelerometer data to compute a proxy for distance. We used that along with the metadata that they provided us because we did want to get their walking course um, specifications. So that's how we did it in the home. And I think that was maybe 10, 20 patients or so that we were able to do that with no prior experience with the technology.
Thank you, Melissa, for clarifying that. Um, okay, so now, I mean, we've been talking a little bit about this, but I want to shift gears and talk about what it takes to trust adopting a new endpoint like cardiac effort in clinical trials. So treatment sensitivity is something that adopters, sponsors care about. So I'd like to ask this group, maybe Dan, Melissa, Dimitri, what is the most compelling evidence that cardiac effort detects treatment effects, and where do we still need more data before drawing, firmer conclusions?
So based on our experience, and we've looked at it in multiple different disease states. We've looked at it after blood transfusion. We've looked at it after recovery after acute pulmonary embolism. We've used it in PAH. We've used it in PH-ILD. And what we've seen is that when we add therapy, heartbeats either decrease, walk distance increases, or you get both. And so you just have more chances to detect improvement, a more subtle change you can find benefit that, because of all the pitfalls we heard about 6 minute walk, may mask or make it more difficult to identify. I think one of our most exciting findings that we found was the benefit of supplemental oxygen in pulmonary arterial hypertension patients. And we had patients who don't routinely wear it. They did a 6 minute walk with heart rate monitoring with 2 liters oxygen flowing by. And then they also did it with a room air tank. And so they didn't know which tank was which. And we had them do 6 minute walk with cardiac effort. And what we found was that in pulmonary arterial hypertension patients, oxygen decreased their cardiac effort, but it didn't change their 6 minute walk distance. So their physiology got better with supplemental oxygen, but their walk distance didn't necessarily change. And then when we gave it to a control group, there was no change in anything. People who clearly didn't need oxygen, heart physiology didn't improve, walk distance didn't improve. We found that it decreases as the heart lungs recover after blood clots. And especially in diseases like pulmonary hypertension, interstitial lung disease, where you've got two or three hits as to why their 6 minute walk may be impaired. Having the ability to detect improvement in physiology during a structured exercise test allows for drug benefit to be identified that may be missed because of underlying interstitial lung disease that's not going to get better. They're more frail or sarcopenic, so therefore their walk distance, they may have more difficulty increasing their walk distance. So it just allows benefit to be seen sooner or when it's difficult for walk distance and isolation to be detected.
Thanks, Stan. We do have some questions in the chat. There is one question around any discussions that you've had with FDA or EMA regarding this measure. Is there something that you're able to share with the group?
Yeah, I actually just responded in the chat, but we did meet with the FDA's DHT steering committee recently, and they had very positive feedback to the research and the work that we presented to them. They have recommended that we pursue the COA qualification program, which we are, and we're currently preparing for a pre-LOI meeting. In parallel, because of some of the topics in that conversation that came up was meaningfulness to the patient. We are also looking at introducing more COAs into our upcoming studies that specifically ask patients um, to object to create a subjective response of cardiac effort. Because some of the other questionnaires don't specifically ask them about the digital measure. And so we're really trying to explore other instruments that we can add to our studies to strengthen the meaningfulness to the patient. We also have a meeting with Galen Research to discuss usage of their CAM4 instrument in upcoming studies.
Great. Thanks, Melissa. And I do see that there is a hand raised, but we will open up to audience question in just a couple of minutes. I have one or two questions left for our panelists. Um, I would like to ask this to everyone on our panel today, but if someone in the audience leaves today and has a special interest in implementing digital measures for cardiopulmonary trials like cardiac effort, what is the single most useful thing that they can do in the short term?
I think it's important to know how your trial is going to be set up, what kind of benefit you're expecting, and depending on what those are, finding a device and team that can help guide and navigate. During the Gossamer phase 2 trial, due to supply chain constraints, they were forced into a device that they didn't want to initially use. And that led to some of what Gabe was talking about, where the device didn't store the data. And so if you lost Bluetooth connection, the data was lost. And so they had some data loss because of technical issues. So if you're thinking about clinical trials, it's really important to reach out to Medidata or whoever about what measures you want to collect, how it's going to be stored, having fail-safe processes in place so data isn't lost. And to me, that's probably the most important thing.
Yeah, I'll add to that we frequently see uh, digital health technologies being written into protocols without a lot of thought into how the data is going to be analyzed. And oftentimes at the end of the study, when we're analyzing the data, there's a lot of protocol changes that we would have liked to have made if we had known exactly, what the analysis plan was going to be like. And so I think first, like really understanding the measures that are important to your study, how you're going to analyze those measures in context with the other data in the study, and then go upstream. How does that influence your protocol design? And then what's the best piece of technology, the best hardware, software companion that is going to meet all those requirements?
Yeah, I fully agree with both Dan and Melissa. I think we need to start with clearly defining the use case scenario. Followed by that, we want to clearly state what the decision a particular measure will support, who's the target population, and how the changes will be interpreted. I think once we do that, then we could lock in the measurement system and identify the evidence gaps that needs to be closed.
I would add just a few remarks. As you saw in the chat, the measure is being always planned to be qualified as a COA. And it's very important to collect sufficient evidence from the patients in the target patient population regarding its meaningfulness and relevance to the patients. And it's a very tricky exercise because if you think of 6-minute walking test, I'm pretty sure if you approach a patient with pulmonary hypertension and you ask that patient, do you actually care about how fast you can walk for 6 minutes or how far you can walk for 6 minutes? I have very strong doubts. I don't think that that patient would really care about it, right? But on the other hand, the regulatory landscape still requires the collection of this type of evidence. Even though the measure can be extremely helpful, indicative, and have the best psychometric properties, it will be required to present evidence coming from the patients regarding its meaningfulness and also regarding what change in that measure is perceived meaningful to the patients, to the agency, or to any other regulator body. And the earlier this work is started, the more evidence can be generated.
Great. Thank you. We have come to that time for audience questions. Um, to engage in this part of the session, feel free to use the raise hand function to ask your question live to the panelists. You can also add some questions in the chat or in the Q&A. So there is a question here that I'll just voice to the panelists, which is how do you anchor changes in cardiac effort to meaningful changes clinically?
So that's where randomized control trials are super helpful. The Gossamer phase 2 trial is currently under review right now. And then we've looked at just single center changes in cardiac effort after adding therapy, as well as in stable patients. And what we found is that cardiac effort, the changes track with changes in emphasis 10, so quality of life metric and pH. And the MCID of 6 had a bigger drop in cardiac effort than less than 6. So single center, we've just observationally looked at it, but really a larger randomized control trial is needed to find bigger anchor signals.
Great, thanks, Dan.
I
Dr.
would
Dimitri, go for it.
to Dan's words. Since the qualification pathway is core, and core by definition is how patient feels alive and function, it's by definition PGI-S and PGI-C, patient global impression of severity and chance, need to be collected to establish the MCID. So again, due to the regulatory landscape, the primary focus on patient feedback and not always or sometimes on physiological signals.
And I'm glad you mentioned that. That's exactly what we're currently speccing out for the upcoming studies.
Great. Any other questions from the audience? Again, feel free to use the raise hand function. There is a question in the Q&A box this time. Do you see a possibility of an offline first approach due to scalability being a major issue, especially in rural areas with low connectivity, as well as in resource constraint settings?
So as part of the study Melissa was talking about, we recruited patients who did not live close to our clinic. We're talking an hour to 2 hours away. And they were able to use the technology and didn't have any issues with it. Multiple, and this is where it's important to know what device you're doing and how it handles it, but a lot of devices will save the data on the hardware, and then you just wait till you have internet, and then it can sync it to the cloud. Wrist-based actigraph, you can, or Garmin, you can save data on a watch, and same thing, send it back, and then you can collect the data later. So there are devices that allows for rural implementation, and then as long as you can get to internet, it shouldn't be an issue.
Thank you, Daniel. It seems that we have a raised hand. Alyssa, do you mind unmuting and letting that participant speak to ask their question? Okay. Okay. As we figure that out, there is another question here, which is, do you have interventions for at-home 6-minute walk tests if the patient pauses because of motivation, not physical capabilities for some days?
So if someone pauses, you're still going to collect heartbeats, and then you still get the total distance. So using accelerometry data and the home unsupervised setting, you can take that raw data and analyze it a couple different ways to come up with a value for how far they walk, steps, laps, distance. if somebody stops, that really hurts the measurement. And if somebody stops, you probably just need 6 minute walk because they're walking 200 meters or less. So if somebody's walking that low, there's other factors going on where you just wonder what's going on with the person. I don't think anyone would feel reassured or things are going great if somebody needs to pause for 30 seconds to a minute during a 6 minute walk test without knowing any other information. If you just told me somebody needed to stop, then that would tell you. Now, what you could look at is what their acceleration heart rate trajectory was, and did they stop because their heart rate was 150, or did they stop when their heart rate was only 100? And then that gets more to the physiology causing stoppage or motivation, person just wanted to stop. Either case, I wouldn't want somebody in my clinical trial who walks for 3 minutes, stops, and their heart rate's only at 95 because of motivation. That's what kills your placebo group.
Yeah,
I'll just add a kind of a tangential observation is that when you do record ECG and accelerometry during the walk, then you have a replayable audit that you can go back and review and provide context to actually what happened, and it can can be used in the clinic for QA as well. So when you're the walks traditionally are not video recorded, and there's typically only one clinician observing the test. And so if there's a mistake with the lap counting or another pause or another interference of the walk, you'll never know in the, in the data report. But with the sensor data, you can replay that and look at that data many times to really try to understand what may have happened.
And to just comment further on what Melissa just said, so in our own data set, doesn't happen commonly, but as we're analyzing cardiac effort, you get a number that just doesn't make sense. And so we've gone back and we've replayed or audited the acceleration data, and study coordinators sometimes miscount laps. And so when you count the laps with the accelerometry data, you're like, are you sure that wasn't X number? And then they go back to their source document and there's an error transmitting what they wrote in their source document into the EDC. And so we found mistakes just by looking at the time-stamped acceleration during the 6 minute walk test. And so if I'm running a clinical trial, having that as like my fail-safe, go back, make sure I'm not getting killed by placebo because somebody miscounted laps is another benefit that we didn't even talk about today, but is included with most of these heart rate monitors with accelerometers that just protects the house.
Those are great points. Okay, we'll try this one more time. Daniel Muzica, you had your hand up. I'm going to ask you to unmute if you'd like to ask your question live. Or otherwise, feel free to put it in the chat as well. There is another question around how your team wrote into the protocol, how to ensure data privacy and security using data at the home.
We followed our IRB protocol. It was de-identified. No patient information was collected. We gave them unique logins. I guess the only thing that now we have to include is that if it's transmitted from their IP address, that people may have access to that. But outside of that, there has not been any institutional review board issues with doing these kind of assessments.
Great. And maybe last question for today. This one is directed to you, Melissa. If you could highlight what were the results or observations for the remote 3 minute walk test at home that you had mentioned earlier.
Yeah, so as Dan mentioned, when you reduce the length of the course because of the turns, you're going to get a shorter walk distance overall. And so we had the participants do a shorter walk in the clinic, and then we had them go home and do it so that we can compare it. They also did the standard walk as well. So we had all of these data points for this particular study. And what we observed with the 3 minutes is that from a cardiac effort standpoint, we do believe that 3 minutes in a shorter duration walk is sufficient to get to a statistically comparable cardiac effort than what was done in the clinic. And so very promising. The hypothesis was that 6 minutes was sort of arbitrary number from decades ago, and based on how the heart rate responds to when you're starting exercise, um, we believe that you can kind of get to the point that you want to be with your heart rate function in a shorter amount of time, and therefore reducing the burden for the patients so they can walk 3 minutes instead of six minutes. And from a cardiac effort standpoint, we're still getting the same data. And Dana, if you want to add any other insight on that.
No, that's really helpful. And then I just want to answer the person who asked about AFib and beta blockers. So we have looked at atrial fibrillation. We have looked at beta blockers. We recently, it just accepted and we published soon, cardiac rehabilitation after heart surgery, cardiac effort measurements. And really the thing that we found is that if someone's on an appropriate beta blocker dose, it doesn't, because that's going to be constant throughout the entire monitoring period, that's not a problem. So patients on beta blockers still can augment their heart rate to low 100s. And what we have found is some patients may be more chronotropically insufficient because of beta blockade that impacts their 6-minute walk. Same thing, we found AFib. So if AFib's uncontrolled, yes, that's a problem and will definitely impact how somebody's functional capacity is. But you can look at that during the 6-minute walk and adjust therapies to know that they may be in AFib, but if their heart rate's targeted less than 120 with activity, that should be a constant over time. And so we haven't found that to be a problem either.
Dimitri, go for it.
I would probably like to add a very simple fact. If some patients take some medication throughout the clinical trial, does it really truly matter? I don't think so, because as long as you're interested in a change in a measure of interest and all other confounding factors remain the same throughout the course of a clinical trial, that means that this factor cannot really influence the change in that measure.
It's a great point. Okay, before we wrap up, any final thoughts or words from our panelists today?
I've really enjoyed the questions. I hope we answered most of them. We do have a lot of publications that may be helpful, so maybe we can send that or share that. And we have very recent publications also, and we're working on more this year. So yes, I think Dime, you'll figure out how to reach out if there's further questions, but just wanted to mention that there's a lot of publicly available information if you want to dig a little deeper into some of the methods that we've used for our research.
Excellent. Yes, we can send a post summary email with the links to the new research. Okay. Because we've been talking a lot about the added value of digital endpoints, I did want to share this helpful resource from Dime, which includes interactive guides for developers, for pharma navigating their DHT endpoint strategy and practical implementation. There's a chat box you can interact with, and we've vetted all of the resources. Um, Before we wrap up, I will put a little plug. At Dime, partnerships are central to our mission of advancing evidence-based digital medicine. We would love to help you amplify your voice through webinars like these, through roundtables, thought leadership, as well as connect you to key stakeholders, regulators, health systems, clinicians shaping this field. If you're interested in learning more, please click the link or scan this QR code here to fill out our Get Involved form and explore partnering with Dime further. And with that, I'd like to thank you all for joining us today, and a special thank you to our speakers and our partners at Metadata. Your work generating clinical evidence and real world data behind endpoints like cardiac effort is exactly the kind of information, the innovation we need to move this field forward. So we're so proud to partner with you and thank you all for joining us today.